Bismillahirrahmanirrahim. Dear viewers, I am Professor Dr. Muhammad Schwann and today we are going to discuss Neuroglia. Now, what are Neuroglia? Neuroglia are basically sporting cells in our nervous system. Okay, so these are the cells which support the major components of the nervous system, that is neurons. For neurons, they provide a sporting network and they have key differences from the neurons. We know that neuron, it is an excitable tissue. It generates nerve impulses and nerve impulses are transmitted in the neuron. While on the other hand, the neuroglia, which are the sporting cells, they are non-excitable and these do not transmit nerve impulses. The number of neuroglia in our nervous system is about 10 to 50 times as high as the neurons. Now, neuroglia can be divided into two major types, that is macroglia and microglia. First of all, we will talk about macroglia. The macroglia can be further subdivided into three types, that is oligodendrocytes, which form myelin sheath in the central nervous system. Then there are Schwann cells, which form myelin sheath in the peripheral nervous system, and we have then astrocytes. So these are the three types of macroglia. I repeat, oligodendrocytes, Schwann cells and astrocytes. Now astrocytes are again of two types, that is these protoplasmic astrocytes which are found in the gray matter and these fibrous astrocytes which are found in the white matter. Now there are differences between these two. The protoplasmic astrocytes, they contact the neurons at the level of synapses. Okay, as you can see here, this protoplasmic astrocyte, it is contacting with the neurons at the level of synapses in the gray matter, while this fibrous astrocyte, it makes contact with the neurons at the nodes of Ranvier. Okay, it is making contact with the neuron or axon at the node of Ranvier. Then in case of protoplasmic astrocyte, its glutamate exposure is high. Its glutamate exposure is high and it has high GLT1 expression. As you can see here, it has high GLT1 expression. While fibrous astrocyte, it has low glutamate exposure and it has low GLT1 expression. Then the third difference is that the protoplasmic astrocyte, it has got high glycogen content, while the fibrous astrocyte has got a low glycogen content. And there is also difference regarding the gap junction coupling. There is intense gap junction coupling in case of protoplasmic astrocyte, as you can see here, while there is low gap junction coupling in case of fibrous astrocyte. So these are the few differences between the protoplasmic astrocyte and fibrous astrocyte. Now regarding the functions of the astrocytes, their major function is that they form blood-brain barrier. Okay, as you can see here, this is an astrocyte and it is forming a blood-brain barrier in the CNS. So it regulates the entry of substances from the blood to the brain tissue. Then these astrocytes, as you can see here, they form a sporting network in the CNS. They form a sporting network in the CNS. They also form a framework along with the developing neurons migrate to their destination during embryogenesis. Then a very important function of the astrocyte is that they regulate the concentration of electrolytes in the CNS and they also regulate the concentration of neurotransmitters in the CNS, especially at the level of synapses. As you can see here, the concentration of glutamate is being regulated by an astrocyte. Then the other form of the glia cells is microglia, and these microglia, basically these are the scavenger cells and these resemble tissue macrophages and they remove debris resulting from injury, infection or any disease. Actually, these microglia, they arise from macrophages outside the CNS. So, in this way, in this short video, we have learned about the function of neuroglia, which are of two major types, that is macroglia and microglia. The macroglia are further of three types, that is oligodendrocytes, Schwann cells and astrocytes, while the microglia, they are the scavenger cells in the CNS. So, hope now you would have understood about the functions of these neuroglia. Please subscribe, like and share this video. Thank you for watching. Allah Hafiz.